Okay, I'll just keep doing that. Okay, good. These are my relevant disclosures. And so this is my challenge. I've got to take actually a 2-hour presentation. Vic says I've got to stay on time, and I've got to compress it down to 15 minutes. So either I have to go Marty Friedlander style, which I know Bart doesn't like, or I've got to take some content out. So I'll probably go the latter strategy. So let me start first with clinical trials for dry AMD. And these trials are largely focused on how we can slow or stop the progression of geographic atrophy. And there's actually a number of trials you should be aware of that I do want to talk about, or at least the general pathways briefly. First of all, one strategy has been to modulate complement, and so there's a number of late-stage trials relevant to that. You can go with anti-inflammatory route. You can do visual cycle modulation. We'll talk briefly about that. There's neuroprotectant options, and finally, there are stem cell-related therapeutics. So I'm going to focus primarily on complement modulating drugs because they're sort of the hot topic, and you might sort of ask the question, well, why target complement? And there's a lot of different rationale. I mean, we see drusen-like deposits in patients, younger patients who have complement-mediated disorders, and there's obviously a lot of genetic data to support that complement linkage. Now, this is sort of the painful slide where people's eyes glaze over, and you have to sort of remember these different pathways from medical school biochemistry in terms of how the complement cascade works. But we know that there are these 3 different pathways that ultimately sort of come down to C3, which then goes on to C5, and then the membrane attack complex. But what's interesting, though, is that actually when we think about this genetic link to AMD, there are a number of different genetic loci associated with AMD where you can see it's red all over this complement pathway. So that's what kind of makes us think complement is somehow relevant. So what are some of the agents that are in some late phase type of trials? There's the anti-C3 intravitreal approach. There's a number of different anti-C5 approaches that different companies have been pursuing. And then finally, there is anti-factor D, which is of course lampalizumab, and that's probably gotten the most press, and it's in a late phase study certainly, and something certainly that we should talk about here. It is a antibody fragment, so it's a Fab or the AB portion of the antibody, and in this case, lampalizumab actually because it works a little bit upstream, it's able to inhibit the alternate pathway and ultimately the production of C3 and C5 through the alternate pathway. So then, if you go back to that really painful complement pathway slide, that's where it sits. And so the idea is here you put a stop here, and then you're able to at least stop that direction from the alternate pathway. And the Mahalo study was a phase 2 study. We were very much involved in this because we were the reading center for the Mahalo phase 2 trial that was looking to see whether this approach could ultimately prevent the progression of atrophy associated with AMD, which is kind of exciting to be involved in this because this was potentially a primary endpoint that was imaging based. And so in any event, the study did meet its primary outcome. They saw that with lampalizumab given monthly seemed to be associated with a slowing of the progression rate of atrophy. But note that the p-value was less than was 0.1. I mean, it was less than 0.2. That was the pre-specified, but it was not 0.05. But they still said it met it because that's what they specified in advance. So in any event, they went on to do some sub-analyses, and they said, okay, it turns out that half of our patients actually also, in addition to having AMD and the like, they also specifically were positive for a polymorphism in the CFI gene. And the reason that turned out to be important was it turned out that patients who were positive for the CFI polymorphism seemed to have a substantially higher growth rate in terms of their atrophy. And so when they looked back at the data and sort of dissected it, looking at these things separately, they found that in fact the effect of the drug seemed to be primarily in those patients in the left-hand panel. So in any event, my point here is that the effect of the treatment primarily seemed to be driven by that CFI positive group, and you can see there was a more substantial reduction in the growth rate of atrophy, and if you're CFI negative, there was like nothing. You saw no effect at all. occur at the level of the photoreceptor, and then that results in an isomerization, and all trans retinol is formed, and that, of course, is recycled in the RPE cells through the action of RPE65 to get it back to 11-cis, and that's how sort of the visual cycle occurs. And it turns out that this RPE65 is quite critical for this type of recycling. But one thing is the cones. It sort of reminds me of Alfredo's escape mechanism, or a way to get down his electron transfer chain through complement, or the component 2 there. It's sort of the same thing. The cones have another mechanism. They can get their 11-cis retinol from Mueller cells also. So an inhibition of RPE65 primarily suppresses rod function. So the nice thing about this kind of approach, unlike lampalizumab, which is an injection, and David Park told us about the challenge we have with so many injections, but this is actually just a pill, so that seems very attractive. And as you might expect, I mean, in the early phase studies, they were able to demonstrate that whereas the rod function, and this middle bar is after treatment, was pretty significantly depressed, and then it recovered, that's the third bar there, after treatment was withdrawn, cone function was largely not affected so badly. So this really seemed to just make patients' night vision, unfortunately, worse, but the atrophy hopefully wasn't progressing. So in any event, this led to a phase 2/3 program, and we were told to expect results this year. And in fact, we got them. I didn't get a chance to update this slide a couple of weeks ago, and it didn't work. So we didn't see an effect on atrophy, and so they're still mulling through the data to see if there's an avenue that they can continue. So that was kind of disappointing. I'm not going to talk about the others, only because I have no time to cover them, or Vic will kill me. So, okay. So moving on to wet AMD, you might ask, well, don't we already have good treatments? Why are we talking about more trials for wet AMD? And the answer here is that, of course, all of our therapeutic strategies so far have really focused on VEGF and really trying to suppress VEGF completely. But there's potentially more to it. I mean, we know that these major anti-VEGF trials have 3 agents that really seem to show quite substantial benefit. But I will say there are still some limitations. Not everyone responds, and you have to treat patients pretty frequently, which can be a real challenge for patients. And in fact, we also know from these trials that if you then follow the patients even longer, there tends to be a reduction in vision over time. Some of that is probably under treatment. Some of that is atrophy progression. There have been some wonderful studies. This is from a good friend of mine, Frank Holtz in Germany. They conducted a study across all of Europe to see what happens to patients long term. And each of these different graphs that you're seeing, or lines you're seeing, are different countries. And basically, it's interesting. The UK patients, I guess, do a lot better. And if you're in Italy, you're really in a world of trouble, it looks like, according to this. But in any event, there's also far fewer treatments being given in Italy compared to in the UK. I don't know, maybe the patients aren't coming back, or they just don't feel like getting an injection. I don't know. But it's very interesting that that was seen in this study. So in any event, the point here is, and this may be one takeaway message, we haven't solved this. There's still a problem here. There's a need. And so there's some potential ways that we could tackle this. One is with better anti-VEGF agents. There's a whole host of options that are in trials or been through trials. I can't talk about all of them. I'll focus on one just because I think it's a really cool strategy and an approach, which is in part because it's not even just this one agent. It's like a class of agents that we can use for a variety of different applications potentially. And that's, of course, the DARPins. And abicipar pegol is the one that's in a phase 3 trial right now. It's the only one I'll talk about for time considerations again. Again, DARPin, what does it stand for? So get used to this name. Design, anchor, and repeat proteins. And it's really a cool way of targeting a variety of different pathways. But it's nice because it's very small in size and it's very stable. So you can put it in the eye, and theoretically it should last a long time. This is a busy slide, but mainly the message here was that it seems to last longer than ranibizumab, at least in sort of in vitro type of assays. But it's went through a phase 2 sort of program, ultimately culminating in a stage 3 phase 2 trial, which ultimately demonstrated there seemed to be this nice dose response curve, improvement in vision with abicipar perhaps given less frequently compared to other treatments, and there were a lot more patients improving many lines of vision compared to the ranibizumab treated patients, so that all looked exciting. These are some of the patients from that trial. But in any event, that led to the Cedar and Sequoia studies, which we are a part of here as well, which will be the definitive studies. And these are some of the entry criteria for putting patients into the trial. So if you have CNV patients, and they're, you might want to consider options which could work very well and maybe give them longer duration of action. I think it's a very interesting drug to think about. So please do refer those patients to us. Now, there's also a lot of work on on better delivery system approaches. There's a whole host of different technologies that I wish I had time to talk about. The only one I'll really say anything about is gene therapy, which Alfredo already introduced the concept, so I thought that's what I'll mention. And just so you know, there's a couple of different approaches. All of them center around using something called soluble FLT, which is basically VEGF receptor that you can put it into the eye and it sops up all the VEGF that's sitting there, and it can be delivered intravitreally or subretinally. Unfortunately, so far, the data from these studies doesn't seem to be working quite as well as we'd hoped, and part of it is getting the viral dosing right. So this is still a work very much in progress. This is just from the AVA 101 trial of the soluble FLT given subretinally, and you can see that there wasn't really, as substantial an effect as they had originally hoped. Okay, and lastly, I'll finish up by saying, I started off with this cartoon that sort of said, well, VEGF is only part of the story. There's all these other places where you can imagine that you could intervene and potentially have some type of beneficial effect. And in fact, there's a whole bunch of factors that have been associated with corneal neovascularization as well. And just so you know, again, I'm not going to talk about all these. I'm going to finish actually just a moment here. But there's a whole bunch of different agents that are in trials right now. And the one that probably everyone is talking about the most, and I think because we're going to hear something later this year at the very latest, the beginning of next year, is of course Favista. And again, I'm not going to talk about the rest due to time. And the concept for this agent is targeting PDGF. And the concept here is that the response of corneal neovascularization to anti-VEGF therapy is influenced by how mature the vessels are. And the hallmark of how mature a vessel is whether it's covered nicely with pericytes. So the idea is if you can do something to whack the pericytes, then you'll be able to get more effect from your anti-VEGF therapy. So it's thought that that combination, that two-punch strategy might work better. So again, this is just to illustrate that pericytes are important for stabilizing vessels. And so in any event, let me skip past in the interest of time here, but people have shown in some preclinical studies that when you remove the pericytes, that seems to increase the sensitivity of the vessels to the effect of anti-angiogenic agents. And so again, if you use anti-VEGF therapy alone, that may not be good enough to cause regression of mature, and this should say mature neovascularization. And wiping out PDGF might block pericyte recruitment to the new vessels, but it shouldn't do anything to the already well-established vessels. And so in any event, that's been developed into this product. This is, again, all these long names to remember. If you want to use the generic name, pegplarnib, so get used to saying that. And in any event, this is just a corneal neovascularization study where they showed that you were able to strip pericytes off of this area of neovascularization, but you didn't affect the mature limbal vessels to show that there was some selectivity there, and there have been good anatomic data from the early studies, and so that led to a phase 2 trial, which showed that there appeared to be actual superiority of the combination of pegplarnib and ranibizumab compared to ranibizumab alone in terms of improving vision. And so the phase 3 is fully enrolled, and so we're expecting results, in a period of months. So in any event, I'll leave you with the thought that there are a whole host of agents on the horizon, and unfortunately, we're probably going to be doing more and more injections. And so hopefully, again, I know this has been a bit of a whirlwind sort of tour, but there's just too much to cover. But advances in our understanding of the pathogenesis and pathophysiology of both dry and wet AMD, I think, have clearly uncovered a wealth of new targets for. Oops. Was that you cutting me off, Vic? No, okay. All right, and so several agents have progressed to large-scale phase 3 clinical trials, and so I think this will hopefully lead to better treatment options for our patients. And so certainly consider referring these patients in. We're really interested in offering these options to